Greetings. So let's talk about the cells that are part of the adaptive immunity. And we are first going to look at cells that are part of the humoral arm of the adaptive immunity. So just to recap, remember immunity is innate, adaptive. Within adaptive, you have the humoral immunity, which is the immunity which helps us deal with exogenous or extracellular uh, debris like bacteria. And then you have the cell-mediated immunity, which helps us deal with endogenous threats like viruses and tumor. Okay, and so today we'll talk about cells that are involved in the humoral adaptive immunity. Okay, now, which are these cells? So remember, we had the antigen presenting cells and antigens, right? Which we got from the innate immunity, and both of them flowed into the lymph channels, right? So that's one. So they are involved. And then we have T helper cells, and these are the CD4 cells. Okay, these have CD4 markers on them. You have the B cells and the plasma cells, and then you have antibodies. So these are the big cells that are involved in the humoral immunity, right? So again, with lymph system, we got our antigens, or we got the macrophage and the dendritic cells. So the macrophages and the dendritic cells gave us what? Gave us a cell, right, with MHC2 and an antigen on it, right? Now, what if, so that we've already gotten in our lymph channel, so it's ready. What else can we get? Well, we can have an antigen that may sneak into the lymph system. So that antigen may then attach itself to a cell called a B cell. So do we know how B cell gets made? Yes, we have an idea. We've studied B cells. Remember the lymphoid lines? Yes. And then these B cells have these receptors, which are B cell receptors. Okay. And it so happens that this antigen fits this B cell receptor perfectly. So what happens when that happens? Well, as antigen gets picked up, there's an MHC2 complex that gets developed within this B cell. And this is an MHC2, again, marker or molecule. And then it actually attaches to the antigen and it gets to the surface. So now it has the MHC2 molecule with an antigen on it. Okay, so it's doing the same thing that the macrophages are doing, which is what? It is now active and it's presenting the antigen on an MHC2 marker on itself. Now remember, they still have the MHC1 marker with the self antigen on it. Remember, all nucleated cells do. Okay. So regardless of whether it's antigen or it's antigen presenting cells, at one point in time, both of them are doing the same thing, which is they are presenting MHC2 antigen on an MHC2. Now, who do they present this to? That's where the fun begins. So let's start on that now. Okay. So again, we had the B cells. We talked a little bit about the B cells, right? And B cells, we talked about macrophages and dendritic cells. And now this is the cell we are at. This has the MHC2 and it's got that foreign antigen on it. Okay? Okay. Now, we have a cell called the T helper cell. So again, T helper cell is lymphoid or myeloid line. If you're not sure, I want you to now review the video. Okay? But essentially now we're in the lymph channel and lymph system, And we have the antigen that got processed by the antigen presenting cell and is now presenting it to the T helper cell. Okay, so that's where we are at. Now, T helper cell has a CD4 marker on it. Now, it makes this cell very special because the CD4 marker hooks in perfectly with this MHC2 molecule. So these two are best of friends. That's why this is also called a CD4 cell. MHC2 tags in perfectly with CD4 cell. Okay. Now, I'm going to just spend a little bit more time on it. There's something called rule of 8, which helps us remember these T cells and CD4 markers and MHCs. Okay? This will make it easier to remember. CD4 T cells are T helper cells. Okay? They attach to MHC2. 4 times 2 is 8. Okay? CD8 cells, which are the T cytotoxic cells, attach to MHC1. 8 times 1 is 8. So the product is always 8. Okay? T helper cells are CD4 cells that connect to MHC2 of the antigen presenting cells. That gives us 8. They then cause the humoral immunity or antibody response, and they are external. Therefore, the external extracellular enemies. T cytotoxic cells are CD8 cells that attach themselves to MHC1. Remember, all nucleated cells have MHC1. Product is 8. This is cell-mediated immunity. And again, it's for intracellular, internal, or endogenous threats. Okay? So this is just something to remember. We're going to review this again when we get to CD8. But this helps you remember which cells are linking to which cells and which immunity bucket do they go into. It's very, very important to remember and keep it straight in our heads what's happening. Okay, so let's go back. You had the antigen presenting cell. Remember that? Antigen presenting cell. And it had MHC2 and the antigen, and it was presenting it. It presented it to the T helper cell, which is the CD4. Remember rules of 8, CD4 and MHC2 hook into each other perfectly. Now, it also has a T cell receptor, just as B cell had a B cell receptor. So what's interesting about the T cell receptor is the same thing about the B cell receptor. One type of receptor can only attach to one kind of antigen. So, here's this antigen presenting cell floating around, and a T helper cell comes around. Its CD4 fits in, but its T cell receptor doesn't fit into this antigen. So, it's not going to fit. It's not going to fit. And the second one comes around, same thing. Third one comes around, same thing. Wait. This fourth one came around, and the T cell receptor fits perfectly. So now it locks into place. The CD4 attaches to MHC2, and the T cell receptor attaches to the antigen. Okay. Now that's what I call lock 1. It's locked and loaded. However, what happens next is so intense that the body wants to make sure that what it's doing is, it wants to make it doubly sure that that's what it wants to do next. So it has an extra fail-safe mechanism in place, which is called a co-stimulation. Okay? It's called a co-stimulation. Here, the B7 receptor on the antigen presenting cell attaches to the CD28 receptor. And this is what I call an insurance policy. Okay? So I had the MHC2 attaching to CD4 and the T cell receptor attaching to the antigen. Now the B7, so that's first stimulation, and then there's a co-stimulation of B7 attaching to CD28. And this now locks and loads it into place. Okay? So what happens when this happens? Well, that naive T helper cell becomes activated and produces interleukin 2. That antigen presenting cell produces interleukin 1. Okay? Guys, see what we did? We locked and loaded, and then once the lock and loading happened, then interleukin 1 and 2 are produced. Interleukin 1 does what? Remember, we've covered it before. We have covered it before. Gives you what? Fever, C-reactive protein, and increased white blood cells. Okay. What does interleukin 2 do? We've covered that as well. Okay. So it helps the T helper cell proliferate itself, make more copies of itself, and also proliferate the B cells that are presenting the antigen to it. Okay? And these are both very specific for the antigen that it is processing or that is being processed. Okay? So what does it do? IL-4 produces T helper cells and B cells that are specific to that antigen we are talking about, and also produces IL-5, which differentiates these B cells into plasma cells. And the plasma cells give us more antibodies. And antibodies are mega awesome. IgM, IgE, IgG, and IgA. We'll talk a bit more about them in just a few seconds. But they are the end product. They are the end product of this interaction. Okay? So again, we'll just go through it again. Regardless of antigen or antigen presenting cells coming into the lymph system, the antigens get processed by the B cells, and the antigen presenting cells are, you know, both of those pathways are doing the same thing, which is you have your cell, antigen presenting cell, which has an MHC2, which has a foreign antigen on it. Now, if the MHC2 and the foreign antigen attaches itself to a specific CD4 T helper cell, and the foreign antigen attaches to the T cell receptor and the CD4 part attaches to MHC2, that's lock 1 in place. That's the first stimulation. The insurance policy, the B7, attaches to CD28. Once this is locked and loaded, interleukin 1 is produced by the macrophage or the antigen presenting cell, and that gives you fever, acute phase proteins, and increased WBCs. Tries to get more help. T helper cell gives you interleukin 2, so it creates more B and T cells that are specific to this antigen, this foreign antigen. Okay? And interleukin 5 that differentiates those B cells that are being produced into plasma cells and then give you antibodies. These antibodies are mega. They're awesome. IgM, IgE, IgG, and IgA. This is how I remember the 4 big ones. Okay? Mega. And they are the end product of this because they help us with taking care of the threat. So how do they do that? Let's talk about it. So antibodies are the main weapons that attack these extracellular threats. And they also persist in our blood, and they can recognize when they see that antigen again. Again, they're end product of the complex we just saw. So how do they work? So here's you have antibodies, and they're against a particular antigen. Because remember, now we are adaptive, okay? And this bacteria has that antigen, and they attach to it, and they crowd it out and cause precipitation. So it's like you're being at work, and somebody's trying to um, do something, maybe good, maybe bad, and then you have all these other people that are blocking or crowding the person, and they'll prevent that person from doing good or bad, and they're basically crowding the bacteria out. So that's one of the ways it does. What about the other one? So here you have a more complex antibody, and it's a pentamer, 5 antibodies that are connected here, and they have these binding sites, and they actually bind to several antigens of several bacteria, and they clump it, and that's agglutination. And we'll talk a little bit more about type 2 hypersensitivity reaction with IgMs, which cause some disorders based on this. But actually, it's a great system because it clumps it out and precipitates out the bacteria and prevents it from moving around. What else? So I have my antibody and I have my bacteria and antigen, and the antibody can attach itself to the antigen. And then remember that complement classical pathway where you had C1, C4, C2, C3, and the C3 broke up, attached to C5, which broke up as well, C6, C7, C8, C9, and then used opsonization, MAC, and chemotaxis. Those are 3 mechanisms we used to destroy this bug. So that's another way it acts. What else? Let's say if it's not a bacteria, let's say if it's an antigen, because remember it's an antigen that can go inside the lymph system as well. Well, the antigen can connect to the antibody, okay? And it can tag it, and then the macrophage can eat it or get rid of it. And again, this is antigen antibody complex. Sometimes it gets deposited in the wrong places, and that gives rise to type 2 hypersensitivity, which we'll talk more about with pathology. And then I think we've touched again on how there's bacteria with antigen, which gets attached to antibody and then attracts the macrophage to eat it. So these are 5 ways where antibodies actually directly attack and manage the extracellular threats. Okay, just very quickly, structure of the antibody. So if you see here, I know we've been kind of looking at it like this, but this is a little bit more detail. Again, this is the area at the bottom which attaches to the specific cells, and then this is sort of a Y, okay, that it forms, okay. But what it is it's actually a molecule. It's a molecule chain here, and then there's 2 molecules that are connected here. And what is in the middle are these binding sites. So each antibody has 2 binding sites or antigen binding sites, and they look differently. They look different. So they could be different. So you may have one antigen, one antibody that, let me just draw it real quick. Okay. So you may have one antibody. Okay. Or you may have okay, and I'm gonna try my best to draw this where it makes perfect sense. So here, if I have a square antigen, it can go and fit in just perfectly. Okay. Or, and let me draw this here. Okay. Now, okay. Sorry, my drawing is not the best. Here, this may be an antigen which may get and this may be an antibody that may attract and attach to something that's a triangle antigen. And so this area is very variable, okay? And it depends on what the antibody is against, which antigen is the antibody against. So again, we're with adaptive immunity, we are now targeting and are specific to what the immune system is, or what the threat is. So we're thinking through what the threats are. Okay. Okay. So let's do a couple of more things before we are done for this lecture. What I wanted to do, there you are, was talk a little bit more about the antibodies individually. So remember we talked about antibodies and we talked about the mega antibodies because they're awesome, right? So you have mega IgM, IgE, IgG, and IgA. So let's talk very quickly about them just so that we have an idea. IgM, I call it, remember, mega Ig miserable. Just remember that we'll talk about it. It's a pentamer, so remember it's 5 antibodies that are connected to each other. It's a primary response. IgE is a monomer, so 1 antibody. A lot of it is in our respiratory or GI mucosa. And remember, IgEs connect with basophils, and you see allergies. If IgEs kind of work with eosinophils, they try to attract the parasitic worms. Okay, IgG. Okay, these are the most abundant antibodies. And most of them are in our blood and blood plasma. And they are our secondary response. So we'll talk a little bit more about it next. Okay. Like I said, they're most abundant. Also, they can cross the placental barrier. So they can be transferred from mother to baby. So it's interesting. You have a baby, and while it's within the womb, mama can give uh, it the adaptive immunity that that she has developed. The baby can have it. Okay, so that's one of the ways that IgGs work. Last but not the least is IgAs. Okay, and they're a dimer. What does di mean? 2. Just like penta is 5, di is 2. So you see 1, 2. Again, they're secreted in body fluids, saliva, sweat, and mother's milk. So IgA, so baby when they are lactating or drinking mother's milk, they can get a lot of IgAs. Now, let's kind of look at a couple of patterns. So I know we talked about increased WBCs, right? So let's just look at some patterns and get some ideas. So let's say if it's IgGs and IgMs, let's say they're elevated and you're seeing neutrophils that are going up. You can think bacterial infection. Okay, these are some general tips. If you have increased B cells, again, increased extracellular threats like bacteria. Okay, increased T cells. So remember, T cells or T lymphocytes are viral. So that's viruses. If you have increased basophils and mast cells and you have increased IgEs, now I'm thinking what? Allergies with histamine. Okay, so that's type 1 hypersensitivity. Okay, what if you have IgEs that are high with eosinophils that are high? Maybe parasites. I think we've already talked about IgEs and basophils and mast cells, and if they're high, I'm thinking more along the lines of allergies. So remember, we had these non-specific signs and symptoms of fever and increased C-reactive protein and increased WBCs. Well, now we can look at white blood cells and we can look at these antibodies and start to see, hey, these are some of the general indications which cause these rises in the cell lines. Now, here we talked about IgGs and IgMs, right? Right? And the fact that they rise. So let's just spend a couple of more seconds, a couple of more minutes on how they rise and fall. This is very, very important, and I want to make sure we are covering this with you because this actually has a lot of implications for clinical medicine. So let me just draw this first. So this is x and y axis. So this is y axis and this is x axis. Okay. And on the x axis, I have the days or the time. And on the y axis, I have the number of antibodies. So what happens is I get exposed to an antigen day 0. Okay. And so I have a little bit of lag time, and then I start to see some antibodies get developed. Okay. So I've been exposed to the antigen the first time. So what happens is, as I am getting exposed to the antigen the first time, IgMs in the first exposure form the quickest. So you see how they formed the quickest? Okay. IgGs, that's denoted by the blue line here. So IgMs are denoted by the red and IgGs are denoted by the blue line. So again, IgMs get to the peak way quickly than IgGs do. Okay, so there's a lag time, and then IgMs get made first, and then IgGs get made first, and then IgGs get made. Okay, and then the threat is neutralized, and everything goes back to 0, and you would still have IgGs that would be circulating. So that's why this would be more along the lines like this. Okay, and let's say you get another exposure of the same antigen. Okay. And so this second exposure, actually the IgMs don't go that high, but the IgGs go very high and they form the quickest. So with first exposure, IgMs form the quickest. With second exposure, IgGs form the quickest. Okay? So the way I think about it is, when we are drawing blood from a patient who's sick and is miserable, M, and has been exposed the first time, you're going to see more IgMs during the miserable phase or the acute phase of the disease. Over a period of time, their symptoms, they become not miserable, they become happier, and they form IgGs. This is evidence of long-term immunity. Okay, so when we are checking our vaccine titers, so we give them vaccines, we'll talk a bit more about it later, and we check the titers. We're checking for titers of IgG. That's why they take a little bit longer to form. That's why after the vaccine, we got to give it a little bit of time and then get blood for IgGs. Just remember, it's going to take a while to get these up. And then we also want to make sure that they don't go down below the protective level, because if they do, then we need to give them a booster. Okay? Because the moment they get exposed to the antigen or the bad guy or the pathogen again, the IgGs are going to suddenly become very, very high. They're going to form very quickly, and they're going to fight that and prevent that pathogen from taking hold. Okay? So again, this graph and understanding this has implications for understanding blood tests, understanding therapeutics or medications or vaccines that we're giving, and also has implications on how we track diseases. So if somebody comes in with a certain infection and you see high IgMs, you can say, they were exposed. This was their first exposure. Their symptoms along with the lab tells me they got exposed to it. After a period of time, if the IgMs go away, the IgGs are more, you can say, yep, this person now has protective titers against this particular disease. So again, let's try to recap and see if we have a better sense of what we covered in this video. So again, we talked about cells of humoral immunity. So this was adaptive humoral immunity against external agents. So we talked about antigens and antigens and antigen-presenting cells like macrophages and dendrites. And essentially, the net result is you get an antigen-presenting cell that gives you an MHC2 marker with an antigen on the MHC2 marker. This MHC2 combines with CD4 T helper cell because 2 times 4 equals 8. And if the antigen and the T cell receptors are the fit, that's when this complex happens, and then there's a co-stimulation or insurance policy that happens. And then there are two things that happen. Interleukin 1 is produced by the antigen presenting cell, which causes increase in fever, acute phase protein, and increased WBCs to recruit more people. And the T helper cell produces interleukin 2, which causes clonal expansion of T helper cells and B cells that are specific to that particular antigen. And then interleukin 5 that differentiates the B cells that were created into plasma cells, which gives us our antibodies. And antibodies are mega, because they're what we are looking for with all of this. And it's the antibodies that either causes precipitation, clumping, activated through complements, antigen antibody complex, and getting eaten through macrophages that take care of the threat. Okay, we talked a little bit about two antigen binding sites and which part is variable in the antibody in terms of the structure. We did a quick overview of different kinds of antibodies, and we talked about mega antibodies. Okay, and we talked about patterns. If there's more IgE and basophils, you're thinking allergies, IgE and eosinophils, parasites. B cells are mostly for extracellular threats. T cells are for internal threats or intracellular threats. And then last but not the least, we looked at something interesting. We looked at the pattern of infection and how the IgGs and the IgMs go up and down. And the take home is IgM is the first to get formed with the first exposure. So again, the patient is usually with the first exposure because they have the lag time and they don't have protective immunity. They're miserable. Okay. And when you take their blood and you check the IgMs, that's if you see that high, you know, yep, this person got exposed and exposed for the first time. But eventually IgGs we are hoping will develop. And then when they do, the second exposure is the IgGs that kick into high gear and gets formed first. So IgGs are evidence of long-term immunity. And so things like titers with vaccine, that's what we are looking at.